Hi everyone, this is Mark again with video 4 in our series on basics of molecular biology and virology. And today, just correcting a few minor mispoints that I said in the last couple of videos. None of them is crucial to any of the most important conclusions, but just a few minor corrections and a little bit more information. So this one will be short. Okay, a couple of just basic science things. I talked about the fact that enveloped viruses mature by binding to the inner surface of the cell membrane and then budding off and eventually being released as intact viral particles with a membrane layer outside that might contain some viral proteins and maybe some cellular proteins also. So that's correct. The only minor correction is that not all enveloped viruses mature through the main cell membrane that separates the inside of the cell from the outside of the cell. There are various other, inside of our cells, there are various other subcompartments that are enclosed inside membranes, like the nucleus, for example, the endoplasmic reticulum, endosomes, lysosomes, peroxisomes, etc. And apparently some enveloped viruses use some of those other membrane systems. And there's at least one virus that, according to one article I read, actually synthesizes its own membrane. I'm not sure how that works. So that's just a fine point about enveloped viruses. But from what I read, the coronaviruses work as far as it's understood, or as far as I understood it, through the external cell membrane, but that's not crucial to this. The other minor correction, I commented on the fact that many proteins, when they're synthesized by the ribosome, by translation, are cleaved to make a mature peptide that's not the full-length initial gene product. And I gave an example of insulin, where insulin is cleaved. So the mature insulin molecule is actually cleaved by proteases. And that's relevant in this context because some viruses, including coronaviruses, encode in their genomes a protease of their own to carry out some specific cleavages on other viral proteins needed for maturation. And so those proteases, being viral products, are amenable to drug targeting if you can come up with something which is specific to that viral encoded protease that doesn't cross-react with all the proteases in the cell. Now that's harder than it sounds, because all proteases go through some there's only a couple of basic biochemical or chemical mechanisms by which proteases in their active site are able to cleave peptide bonds and chop up other proteins. And so at a most basic level, there's only a couple of fundamental protease mechanisms, and the viral proteases are in some of those categories. So it's tricky to inhibit a particular protease versus all the proteases in the cell, but it can be done. And the refinement is that I said that the insulin molecule, the insulin hormone, when it was matured, was one of those protease cleaved fragments, and it's actually two of them. So the initial insulin protein folds up, and then it makes an internal cross-linking, and then there's some cleavage events. And so the mature insulin molecule actually has two protein subfragments linked together chemically, not one. A fine point. No, actually, that reminded me of something else I was going to say. Okay, and I've lost it. Okay, never mind. All right, so the Solidarity trial I talked about in some detail is a trial by the World Health Organization to test a couple of drugs. And I didn't want to give the impression that those are the only drugs that are being tested. There's one story I saw said that there were over 70 drugs that had potential, drugs already either in use or already partway through testing processes that could have the potential. And so that number is obviously soft. There could be more, there could be a few less, but there's lots of drugs that can be tested. And just because they're not immediately part of the Solidarity trial the World Health Organization itself was rather ambiguous or vague about their details, so probably they want to leave it open, and maybe they'll try other drugs, or maybe if one of the drugs they're testing quickly shows that it doesn't work, they'll switch to another one, et cetera. And it's not the only trial going on, so I didn't want to give the impression that there's only a couple drugs being tried. Many, many drugs are being tried in many places. Interestingly, the Solidarity study from the World Health Organization site involves 10 countries, at least, for now, so maybe greater than or equal to, but the United States is not one of them, and the United Kingdom is not one of them, and it's, given how significant the research establishments are in those two countries, it's rather a notable lapse, and nobody's commented on it, and I have my thoughts about it. Maybe they just aren't interested because they're so big they're doing it on their own. Anyway, it doesn't matter, but yay, Canada is one of the countries in there, and if you go to the World Health site, you can find out what all 10 of the studies of the countries that were involved. Okay, in terms of specific drugs, I mentioned remdesivir, which to me seemed like the most promising because it's an inhibitor. It was designed as an inhibitor of the RNA-dependent RNA polymerase, but I made a mistake. It was not designed against SARS. It was originally designed against Ebola and Marburg, and there are other RNA viruses. But they were shown, at least in lab studies, to work against SARS and MERS. So when I said that that there was evidence that they might work against this coronavirus, SARS-CoV-2, that's true. But they weren't originally designed against that one, a minor point. Another significant thing is I did a little more reading on chloroquine. And as I said, it inhibits a couple of cellular processes. And one of the things that it does, and it's not clear which of all of them are directly relevant for its effects on malaria. But one of them is that it plays a role apparently it lets zinc into cells. So it raises intracellular zinc levels, apparently. And there's some basic studies that showed that the RNA-dependent RNA polymerases and possibly some other viral activities can be inhibited by zinc. So there actually are some studies in the literature showing in cell culture models that chloroquine could inhibit other coronaviruses. Not this one, it's too soon for that, I guess. So it's not totally impossible. However, this still falls, I would say, into the category of blocking a cellular or modulating a cellular process, as opposed to targeting the virus enzymes directly. And we have lots of enzymes in our bodies lots of enzymes in our cells that depend on zinc also. So it's not very specific. It might work, but I would say it falls into the category of things where there's going to be a possibility of toxicity. And in fact, there was just a story in the newspaper today that there was a couple that actually, or one person died and one person's very sick from taking a fatal overdose of chloroquine, which the story said it can be found in some aquarium reagents or cleaning reagents or some disinfectants or something. So I don't know anything about that. But the point is that there's some evidence that it might work, but clearly it's going to be tricky because it would have some toxicity. And people should absolutely not be downing chloroquine hoping that it will help them. We just don't know yet. Okay. The last thing I wanted to mention was, you might think, well, why go through all the trouble when it comes to making vaccines? Why go to all the trouble of making a vaccine in people? Why not just make antibodies in a mouse and inject them into people? So why not make therapeutic antibodies? Because mice generally live faster than us, and rabbits too, and generally their metabolism is faster, and it's faster to immunize them. Not hugely faster, but it is faster. I have done that in the laboratory, and I know a lot of papers on that because I use a lot of antibodies in my research work. So why not make viral proteins? So why not make a viral protein, inject it into mice, and then take out their antibodies and inject them into us, and their antibodies would protect us. It's a good thought. It's not that it can't be done. The problem is that our immune systems would react against the mouse antibody because we would see the mouse antibody proteins as foreign proteins and quickly attack them, and we're actually primed for that for various reasons. So that would be a rapid attack, and that would basically be like giving somebody who was type A, giving them type B blood. If somebody was A negative and you gave them B positive blood, that would be terrible because an A minus person would have antibodies. So A alpha is often for antibodies. They would have anti-B antibodies, and they would also have anti-RH antibodies. There it is. Sorry. So B plus means the person's got B antigens, and plus means he's got RH antigens. And because A minus is minus, he doesn't have protection. And so anyway, that would be really bad. So it would be the equivalent of killing somebody with blood transfusion, like a blood transfusion. So you can't just give people antibodies against something. Now, there may be situations where it's done maybe for the tetanus or some snake venoms. It might be that you can do small enough doses or do it briefly enough if the toxin if it's an immediate toxin like a snake venom, maybe you can clear a foreign antibody could clear the snake venom before you would clear the foreign antibody. I'm not sure about that. But clearly this would require, for vaccinating against a virus that's going to grow, if you were going to protect somebody, you'd have to give them lots of the foreign antibody for a long time. So you can't just do that. But for some years, researchers and therapeutics companies have been trying to do something called humanizing antibodies. And the idea is that these would be antibody molecules that could be grown in a model system, either in mice or in cells in culture. And they would look human enough that if you injected those antibodies into a person, our immune system wouldn't try to kill it, wouldn't inactivate them. So those exist, and it's possible, but you still have to have an antibody developed, and so there's not a humanized antibody sitting around ready to shoot into people immediately against SARS-CoV-2. That has to be developed also, and there's some companies working on that. Most of the humanizing antibodies start with a mouse antibody molecule and change. We know enough about the mouse antibody molecules to know the parts of the mouse antibody molecules that our immune system attacks. So if you can change, delta I use for change, if you can change the relevant parts of the protein so that it still works like an antibody molecule, but it's no longer seen by us as a mouse antibody molecule, but we fooled our immune system into thinking it's a human protein, then that could work. But you still have to raise such a molecule that would inactivate SARS-CoV-2. So that still has to be done, and that's being worked on. So, and as just a minor fine point on top of that, an interesting note is that in us and in mice, antibody molecules are rather complicated. They actually have 4 protein chains. They have 2 heavy chains and 2 light chains. I made a schematic of this earlier that was really poorly drawn. So there's actually 4 different proteins combining together. And there are examples of some animals that have antibody molecules, but they're chemically simpler. It's a single chain or just 2 chains. And so it's easier to engineer them, to humanize them, but they haven't been worked on as much. And the interesting thing is that those are all animals, well, for the moment, they're animals in the camelid family. So camels, llamas, and alpacas. So there are ongoing research and development programs to try to develop humanized antibodies starting with camel or llama or alpaca genes, simply because the biochemistry or the structure of the antibody molecule in these species is simpler, so there's less changes, there's less work you have to do to humanize it. And I'll just point out and make a plea for basic research that if we didn't spend a certain amount of money studying the biology of all kinds of organisms that don't seem obviously relevant for human health, I mean, yes, camels and llamas are important animals for human economy, agriculture, for example, but that doesn't mean we should necessarily be sequencing the camel genome or the llama genome, and why bother? And the answer is the biological world is so diverse, and so much of it has spent so much of its time competing with each other that there's lots and lots of things in the biological world, in the biodiverse world, that are of potential utility to us directly. So you don't have to, in my opinion, actually trying to spend increasing research budgets to study non-obvious organisms is actually not just basic research. It's long-range applied research, and it can be justified on the grounds of enlightened self-interest, even if you didn't think that the world would be a better place if there were more different kinds of plants and animals in it, which I do. But even if you didn't believe that, a world in which there are only people, that's A, that wouldn't be possible, and B, we would lose a lot of potential resources for things that could help us economically or medically. Just one more fine point, one more refinement about antibodies. I noted, or I commented yesterday, that if the virus is a protein capsid, or if it's got an envelope and it's got proteins embedded in the membrane and antibody molecules sit down on it, I mentioned that these antibody molecules can coat the virus to the point where it's blocked and it can no longer bind to a cell. That's one thing antibody molecules can do, but we have, in our immune system, we also have cells that recognize antibody molecules that are attached to foreign objects, and then they eliminate them. So an antibody could be floating around, a viral particle could be floating around, and a bunch of antibody molecules bind to it, and then a cell comes along, or it bounces around in the blood, and if it bounces into a cell that recognizes that this is not just a floating antibody, but it's an antibody molecule that's got something stuck to it that's bad, the cell can engulf that and it can digest it. So, foreign objects, foreign proteins that are, or other foreign biomolecules that become attached to antibody molecules can be inactivated through multiple mechanisms, simply physical obstruction, but also being deactivated by our cells, by cells in our immune system. Okay, that's it for this video. I'll end with a caveat that I'm not officially trained as, I'm not trained as explicitly as a virologist or immunologist. I'm giving you very basic information about these things. I'm trained as a basic researcher, and for the last 10 years I've been doing a lot of human genomics. And in the course of that work I've had to learn about a lot of different human physiological systems, because we'll be doing a genetic study and we'll find that a gene that causes some genetic disorder, it plays a role in some physiological process that I hadn't studied before, so I have to learn something about it. But this is at the level of the virology and immunology that I'm talking about today. This is pretty basic stuff. So if you want to learn more, you'd have to go to a real virologist. Okay, thanks guys.